Okay, so we're going to review the process of DNA replication. And all life depends on the fact that DNA has the ability to replicate and replicate identically. Um, you learn in grade 10 and in grade 11 about the stages in the cell cycle. And DNA replication occurs here in the S phase. And in general, DNA replication and all of the processes we're going to learn about this unit can be broken down into three main phases: initiation, elongation, and termination. So during the initiation of DNA replication, the DNA double helix unwinds at a region called the origin of replication, or the ORI. And that is a very specific nucleotide sequence. In prokaryotes, there's only one origin of replication. In eukaryotes, there are thousands of origins of replication all up and down the different chromosomes. And upon the unwinding of the double helix, initiator proteins will bind to the DNA. So helicase is the enzyme that actually cleaves the hydrogen bonds and opens up the double helix. Single-stranded binding proteins then follow in along behind the helicase to stabilize the unwind strands. And topoisomerase 2, or DNA gyrase, is involved ahead of the replication fork in order to relieve strain on the double helix that's generated from unwinding. So if you think about if you sort of twist two strings together and then unzip them, like open them up, pull them tight, you'll get like a super coiling or a super tight coil down at the base where you're opening them up to relieve that pressure and cause or prevent breaks from happening at random points in the double helix. The topoisomerase nicks in strategic spots, which can then be repaired. So we call that area that's opened up a replication fork, but really it's like a bubble in the double helix. So you can see this is the sort of bubble in the double helix, and the replication fork is opening this way. So this would be DNA helicase, and these are single-stranded binding proteins. During the elongation phase, the first thing that happens is an RNA polymerase called primase moves into the replication fork and lays down a short RNA primer. And that's because the DNA polymerase that catalyzes the reaction, DNA polymerase 3, can't add something to nothing. So it can't start plunking down DNA nucleotides by itself randomly in that replication fork. It needs to be connecting those nucleotides to something. So it connects the nucleotides to an RNA primer, which can be laid down by primase and is then removed. So here, my primase comes in and lays down this RNA primer into the beginning of the replication fork. Once the primer is laid down, DNA polymerase 3 can then add new nucleotides to that primer. But it can only add new nucleotides in the 5' to 3' direction. So it can only add a phosphate to an OH that's there. So we say that the DNA runs anti-parallel. So one strand can be synthesized continuously as the replication fork opens, and that's this one. So DNA has to be made in this direction. So the RNA primer is laid down, and DNA polymerase 3 is going to lay down new nucleotides, and helicase is going to continue opening up in this direction. So this DNA polymerase 3 can consistently work and work and work, and as it gets closer to the fork, that's fine because helicase is going to keep moving the fork down this way in the chromosome. We need another strategy for this strand because this strand is going to need to be built this way. So it ends up needing to be synthesized discontinuously. Basically, as the fork opens up, on our RNA primer is laid down, it's going to be built. the fork's going to open up a little more, a new primer is going to be laid down, and a strand is going to be built. So it's built in discontinuous chunks known as Okazaki fragments. So here is my DNA helicase opening up the fork. Here's my leading strand. So DNA polymerase 3 is able to kind of continuously add and add and add and add into that strand. These are my Okazaki fragments. So here an RNA primer has been laid down and DNA polymerase 3 has used it as a template to build a new strand. An RNA primer has been laid down, DNA polymerase has used it as a template to build a new strand. Here, another RNA primer will be laid down here, and the strand will be built up until this next RNA primer, and so on. So as the helicase opens up the fork, a primer is laid down, a short chunk is made, and then the replication fork will be moved down the DNA some more, a new RNA primer, and another short chunk. And then we have to remove those RNA primers on both strands because we don't want them to be a weak spot in the DNA. So that's accomplished by DNA polymerase 1. And the space is filled also by POL1. And then the fragments, the Okazaki fragments, are joined by DNA ligase. And when the synthesis of the new DNA strands is complete, the two new DNA molecules separate from each other and the replication machinery is dismantled. So I am going to show you an animation. Sounds a volume. Oh my god, how many slideshows have I got open? Okay. So here is my DNA helicase opening up a section of the DNA. So this is a super simplified animation. And DNA polymerase, here this shows you the leading strand is synthesized continuously into the chain. So we're missing our single-stranded binding proteins in this particular animation. So the leading strand is synthesized continuously into this growing replication fork. The lagging strand can't be. So what happens is you have to have a primer laid down here, and DNA polymerase is going to come and build the new strand. And here we're going to have a primer laid down. So primase is going to come, it's going to lay down a short primer, and DNA polymerase 3 can then come and use that primer to start and build some DNA. We'll have a new primer and a new stretch of DNA, and so on. So DNA polymerase 1 then comes in, removes the primer, replaces it, but you can see we've still got a nick in this double helix that needs to be joined, and that is accomplished by DNA ligase. So again, new primase, pull 3, DNA polymerase 1 removes the primer, ligase seals, and so on. And the replication fork continues to work its way down the DNA strand. So here are my important enzymes: helicase, primase, the single-stranded binding proteins, topoisomerase 2 or DNA gyrase, basically the same function. DNA polymerase 1, 2, and 3. So 1 is the proofreader / primer remover. 3 does kind of the bulk of the work, the heavy lifting and replication. 2, we're not sure of. It has some proofreading ability. And then DNA ligase is in charge of joining the ends of those Okazaki fragments. Errors during DNA replication have to be repaired, and they're naturally going to happen with a rate of about 1 in a billion. Usually those are mispairing of bases, or they could be strand slippage. And it's important that, here's a strand slippage error, it's important that those errors be repaired and repaired promptly, because otherwise you're going to end up with a non-functional protein being made due to a mutation. So POL1 and POL2 are the proofreading enzymes. They recognize and correct errors, and that repairs about 99% of the mismatches. So mismatches are rare from the get-go, but then about 99% of those mismatches are repaired by POL1 and POL2. Some are also repaired by additional mismatch enzymes, but anything that eludes those two sort of methods ends up being passed on to the next generation as a mutation. And here compares DNA replication in prokaryotes and eukaryotes. So they both require an ORI. They both occur in the 5' to 3' direction. They both involve leading strands and lagging strands with Okazaki fragments, and they both use DNA polymerases. However, rate of replication is much faster in prokaryotes. There's only a single ORI, but there's multiple ORIs in eukaryotic cells. The DNA polymerase enzymes that are used are different in structure and number. That's the origin. And then linear chromosomes have telomeres, which are highly repetitive sequence at the ends of the chromosome, and their purpose is to ensure that genetic material isn't lost. So because of the way the lagging strand replicates, a little bit of the end of each chromosome is going to get lost. So we have these big, long telomeres that are basically, I want to say junk DNA, but they're not junk. They serve a purpose. Those telomeres are there, and the role of the telomere basically is so that nothing important is going to be lost. And they have implications in aging and in cancer, because as we get into the genes that are being protected by those telomerases, that's one of the ways kind of we age. And Dolly the sheep is a good example of that.